Next, we are going to discuss is the oral mucous membrane. The moist lining of the oral cavity, it is known as the oral mucosa or the oral mucous membrane. What is the function of oral mucosa? The function of oral mucosa is protection, sensation, and secretion. So the function of number one function is protection. It separates and protects the deeper tissue and organs from the mechanical stress, forces, and the external environment. Sensation like temperature, touch, and pain sensation. The tongue has taste sensation, reflexes like swelling, gagging, salivation. They are initiated by receptors in the oral mucosa. Secretion. The saliva it is secreted by salivary glands and contributes to maintenance of moist surface. Thermal regulation. In some animals like dog, the considerable body heat it is dissipated through the oral mucosa by the effect known as panting. So these animals, for these animals, the oral mucosa play a major role in regulating of the body temperature. Drugs like nitrates they are absorbed from the sublingual region. Gingiva and lip mucosa enhance the facial aesthetic form. So all these are the function of the oral mucous membrane. The oral mucous membrane it can be divided into three main types. Number one is the masticatory mucosa, that is the keratinized mucosa. The example is gingiva and the hard palate. So this mucosa, the masticatory mucosa does not stretch and it is attached to the bone. So during the mastication, it will bear the chewing forces and it constitutes the 25% of the total mucosa. The second is the lining mucosa, that is the non-keratinized mucosa. It can be firmly attached or loosely attached. The firmly attached to the soft palate, lip, cheek, and the ventral mucosa, while it can be loosely attached to the floor of mouth, vestibule, and the alveolar mucosa. So the lining mucosa it is exposed to very mild forces. It adapts itself to the movement of cheek, lips, and tongue. So it constitutes the 60% of the total oral mucosa. Third is the specialized or the sensory mucosa. It bears the taste buds and specialized mucosa that covers the dorsum of the tongue including the taste buds. So it will constitute the 15% of the oral mucosa. What are the coming to the histology of the oral mucosa? What are the components of the oral mucosa? The oral mucous membrane it consists of two layers. One is the epithelium layer and second is the connective tissue. The connective tissue components they are the lamina propria and the submucosa. The oral epithelium it is made up of stratified squamous epithelium. Underlying there is connective tissue that is known as lamina propria. The interface between the epithelium and connective tissue is the irregular and upward projection of the connective tissue, that are the connective tissue papilla. The structure interface between the epithelium and connective tissue is the basement membrane. First comes is the oral epithelium. The oral epithelium it forms the primary barrier between the oral environment and the deeper tissue. The oral epithelium it is ectodermal in origin, except the tongue which is endodermal in origin, and it is a stratified squamous epithelium. The cells they may vary from cuboidal to flat squamous cells. The epithelium may be keratinized or non-keratinized depending upon the functional requirement. The keratinized epithelium may either be orthokeratinized or parakeratinized. The epithelium cells they are known as keratinocytes as they contain within the cells fine filament that are known as the tonofilament. So these fine filaments, these are the tonofilaments. These are the fibrous protein synthesized by the ribosomes. When the tonofilament forms the bundle of the filament, they are known as tonofibrils. The tonofilaments will attach to an thickening known as attachment plaque, which in turn adhere to the oval or circular area of the adjacent cell membrane, that are known as the desmosomes. Remember, the epithelial cells they attach to each other by desmosomes, but the attachment of epithelial cells to the connective tissue It is by hemidesmosomes. The basement lamina will separate the epithelium and the lamina propria. It has upper clear layer that is known as lamina lucida, and in contact with the basal cell of the epithelium, and the lower denser layer is known as the lamina densa, that is in contact with the reticle layer of the lamina propria. The cells of basal cells, they are connective to the lamina lucida by hemidesmosomes. Remember the turnover time. It is the time that is necessary to replace all the cells in the epithelium. The turnover time for the gingiva, it is 41 to 57 days, whereas for the cheek it is 25 days. Remember the non-keratinized buccal epithelium, it has a faster turnover than the keratinized gingival epithelium. The basal lamina, remember the basal lamina, it is evident only at the electron microscope level, and it is epithelial in origin. It is evident at the junction of epithelium and the lamina propria. Whereas the basement membrane, it may be evident at the light microscope within the connective tissue. The basement membrane is a acellular structure. It contains reticulin fibers as well as encouraging fibrils. In the skin, it contains fibronectin, laminin, heparin sulfate, type 4 collagen, etc. So the lamina lucida, the basal lamina, it is made up of clear zone and known as lamina lucida just below the epithelial cells. A dark zone known as lamina densa, it is present below the lamina lucida and adjacent to the connective tissue. The lamina lucida, it will contain type 4 collagen. The lamina densa, it is made up of type 4 collagen. Now the layer of epithelium that may be keratinized, non-keratinized, keratinized can be orthokeratinized or parakeratinized. In orthokeratinized, the process of keratinized is absolutely complete within the formation of acellular and anuclear keratin layers on the surface, whereas in the parakeratinized, the cells of the superficial most layer show keratin formation. The cells retain the flattened to oval shaped nucleus. So remember the epithelium of gingiva and hide palate, it is keratinized, while some gingival tissue may be parakeratinized. The non-keratinized tissue, they are the inner lining of the cheek, facial pillar, sublingual tissue, and the floor of the mouth. As we discussed at the oral epithelium, they can be keratinized or the non-keratinized. The keratinized epithelium will consist of following zone. Number 1 is the basal layer, that is the stratum basale. Second is the prickle cell layer, that is stratum spinosum. Third is the granular layer or stratum granulosum, and fourth is the conified layer or stratum corneum. The first layer is the stratum basale layer. It is present above the basement membrane. The cells are cuboidal or columnar in nature. The cells of the basal layer will show most mitotic activity. So this layer is known as germinative layer. As the cells of this layer are capable of division. The cell of the basal layer will synthesize DNA and undergo mitosis, providing new cells. So they are attached to the basement membrane by hemidosomal junction. The second layer is the stratum spinosum, that is arranged in several rows. The cells are larger, elliptical or spherical in shape. The stratum spinosum is known as prickle layer because the cells will show prickle-like appearance. The cells are irregular, polyhedral, and these are larger than the basal cells. The cells, they are joined by intercellular bridges. So they contain glycolipids originating from the Golgi apparatus. Remember, the basal cells and the prickle cells, they together constitute the half to two-thirds of the thickness of the epithelium. The third layer is the stratum granulosum or the granular layer. So this layer will contain flatter or wider cells. So these cells, they are larger than the spinosum cells. The cells of the granulosum, they are flat and are found in a layer of 3 to 5 cells thick. So this layer is prominent in the keratinized epithelium and it is absent in the non-keratinized epithelium. So the cells will have granules. They are creatinine granules in their cytoplasm. So these granules, keratohyalin granules, will help to form the matrix of the keratin fibers found in the superficial layer. The nucleus here in this cell will show sign of degeneration and pycnosis. Podland bodies or keratinosomes, they are the membrane coating granules. They are found in the upper spinous and the granular layer. They originate from the Golgi body system. In the keratinized epithelium, the granules, they are elongated, and in non-keratinized epithelium, they are circular. The last is the stratum corneum layer. Here the cells are flat divide of nucleus and full of keratin filament. So the cells are usually dehydrated. In case of orthokeratinized layer, there is no retention of nuclei. In parakeratinized, there may be retention of the nuclei. The non-keratinized epithelium consists of stratum basale, stratum intermedium, and stratum superficium. The stratum basale, that layer is similar to that of keratinized epithelium. Only the difference is that the cells of this layer in non-keratinized epithelium, they are slightly larger than that of keratinized epithelium. The intercellular bridges, they are less conspicuous. In the stratum intermedium, glycogen is present. Rarely keratohyalin granules are visible at this level, but they are not associated with tonofilaments. The stratum superficial, the cells will appear slightly more flattened than other layers. They contain dispersed tonophils and nuclei and dehydrated cells. This is the structure representing the keratinized and the non-keratinized mucosa. So in the non-keratinized epithelium, there are 4 layers, but in place of granular layer and cornified layer, there are intermediate layer and that is stratum intermedium, and the surface cell layer, that is stratum superficial. Now let us discuss about the connective tissue. The lamina propria is the connective tissue supporting the oral epithelium. The lamina propria, it is divided into 2 parts, the superficial papillary portion and the deeper, that is the reticular portion. The papillary portion, it is associated with epithelial ridges. The papillary layer consists of finger-like projection of connective tissue. The reticular portion is named because of reticular fibers. So the papillary portion, it is the projection of the connective tissue into the epithelium. Below this layer is the reticular portion that contains mainly collagen fibers, blood capillaries, etc. The submucosa, it is a connective tissue of variable thickness. It serves primarily as attachment for the lamina propria to underlying bone or the muscles. The submucosa will contain glands, adipose tissue, vascular and neural components. The keratinocytes, they constitute the major part of the epithelial layer. These cells, they are arranged in different layers. During maturation, they either change to keratin or share in keratin formation. The non-keratinocytes, they are present in both the keratinized and non-keratinized epithelium. They appear as clear cells by ordinary eosin and eosin stain. They need special stain. They are present as scattered cells. The cytoplasm is free of trolofilaments. They do not play any role in synthesis of keratohyalin granules or keratin. The Langerhans cells, they are found in the stratum spinosum and occasionally in the stratum basale. So these have a long thin extension of cytoplasmic membrane that are dendrites. They can be distinguished from keratinocytes by absence of desmosomes and tonofilaments. The important is that these Langerhans cells will contain Birbeck granules. So they contains the rod shape or racket shaped Purbeck granules that allow for identification at the ultrastructural level. For your entrance exam point of view, you have to remember these cells contain Birbeck granules. Their function is they are antigen presenting cells. They will engulf antigen from the external environment, and the intracellular lysosomes they split the antigen into peptide components. So these fragments they are then transferred to T lymphocytes. Merkel cells, where are Merkel cells located? They are situated in the basal layer of the gingival epithelium. So their main function is they serve as a touch receptors. They are occasionally found in groups or cluster. Melanocytes, they are the melanin producing cells. They are located in the basal layer of gingival epithelium. So these cells will arise from the neural crest. They are highly dendritic in nature. The most characteristic feature of melanocytes is the melanosomes granules that is found within the cytoplasm. Lymphocytes, they are associated with the inflammatory response in the oral mucosa. The keratinized mucosa or the masticatory mucosa. So during the mastication, the part of oral mucosa are subjected to force and pressure. So that is why it is known as masticatory mucosa. It is seen in gingiva and hard palate. So the gingiva it is a masticatory mucosa and covers the alveolar process of the jaw. The gingiva it is divided into three types. The free gingiva, this one is the free or unattached or the marginal gingiva. This one is the attached gingiva. And third is the interdental papilla. The free gingiva or unattached gingiva, it is that part of oral mucosa that surrounds the neck of teeth. It is differentiated apically from the attached gingiva by free gingival groove. The free gingiva is composed of stratified squamous epithelium that may be keratinized, parakeratinized, or non-keratinized. The attached gingiva it lies between the free gingival groove and the alveolar mucosa. The junction of attached gingiva and the alveolar mucosa it is known as mucogingival junction. In the healthy mouth, the attached gingiva will show stippling, that is orange peel appearance, that is characteristic of this type of mucosa. The interdental papilla, it extends between two teeth up to the contact point and it is known as interdental papilla. It has a facial and a lingual site. The margins are concave. Due to the inflammation, the interdental papilla loses its concavity. The cole, it is connecting the cole is the connecting facial and the lingual side of the interdental papilla on the proximal side. Its concave shape means the gingiva is healthy. It become dome shaped in gingival recession and inflammation. It is covered by non-keratinized squamous epithelium. Four types of gingival fibers are there: dentogingival, alveologingival, circular, and dentoperiosteal fibers. The gingiva it is made up of epithelial and connective tissue. The gingival epithelium has three parts: oral epithelium, sulcular epithelium, and the junctional epithelium. The oral epithelium faces the oral cavity. It is parakeratinized, show retipex, and connective tissue papilla. The sulcular epithelium it faces the tooth without contacting it. It is thin, non-keratinized, stratified squamous epithelium that extends from the coronal end of the junctional epithelium to the crest of gingival margin. It acts as semi-permeable membrane. The junctional epithelium it provides contact between the gingiva and the tooth. It is made up of stratified squamous non-keratinized epithelium. It is 3 to 4 cells stay thick in early life and the cells that keep on increasing to 10 to 20. It is derived from reduced enamel epithelium. It is attached to tooth by inner basal lamina and attached to the gingival connective tissue by outer basal lamina. The hard palate, it is keratinized masticatory mucosa. It is pink in color and it is firmly attached to the underlying mucosa. So it has a median palatal raphe that extends from the palatine papilla. It is pear shaped and it is formed of dense connective tissue that contains the oral part of nasopalatine duct. So this duct it is lined by pseudostratified columnar epithelium that is will be rich in goblet cells. There are transverse fold called palatine rugae that forms the dense connective tissue covered by epithelium. It has anterolateral fatty zone that is present between palatine raphe and gingiva and the posterolateral glandular area between raphe and the gingiva. Coming to the non-keratinized mucosa, that is the lining mucosa, it is present in the area that are not subjected to high level of friction. It can be firmly attached to the underlying muscles like soft palate, lips, cheek, ventral surface of tongue, or can be loosely attached to the underlying structures like floor, vestibule, and the alveolar mucosa. It is thicker than the masticatory mucosa, non-keratinized mucosa. The lamina propria is thicker with fewer collagen fibers. So this is example of the epithelium of the lip and cheek, in alveolar mucosa, soft palate, and floor of mouth. The epithelium, lamina propria, and the submucosa. Coming to the specialized mucosa. The specialized mucosa is the dorsal surface of the tongue. The different papilla that are found on the dorsal surface of tongue, they are filiform, fungiform, circumvallate, and foliate. The filiform papilla that forms with no taste bud. The connective tissue, it is covered by thick keratinized epithelium that gives a velvety appearance of the tongue. The fungiform papilla, they are numerous, present at the tip of the tongue. They are mushroom shaped, highly vascular connective tissue and thin non-keratinized epithelium. Circumvallate papilla, they are 8 to 12 in numbers, present anterior to the sulcus terminalis. Connective tissue core that is covered by keratinized epithelium. It has taste buds. It is surrounded by deep circular groove into which ducts of von Ebner salivary glands open. The foliate papilla, they are located in furrows along the posterior side of tongue. They may be lined by taste bud. They are not prominent in the human beings. The velvet papilla is for bitter taste. Fungiform papilla gives sweet and salty taste. The foliate papilla, sour taste. Coming to the taste buds. The taste buds, they are present in all tongue papilla except the filiform papilla, present in soft palate and posterior surface of the epiglottis. So each taste bud, it is composed of 3 modified epithelial cells, the outer spotting cells that are arranged like layer of onion. The inner spotting cells, they are shorter and rod shaped. The taste cells contains neuroepithelium. They are 11 to 12 in number. So these cells are slender with dark stained nuclei. The lingual tonsils, they are covered by stratified squamous non-keratinized epithelium. The lymphatic tissues surrounds the crypts and are composed of germinal center and lymphatic tissue that fills the spaces between these centers. What are the age changes in the oral mucosa? With the age, the epithelium appears thinner histologically. There is flattening of the epithelial ridges, reduction in number of the papilla. Langerhans cells become fewer with age. leading to decrease in cell mediated immunity, decrease in the collagen with increase in age. The 4-die spots they increases with age. Elderly women will have symptoms like dryness of mouth, burning sensation, along with the abnormal taste. So the oral mucosa it can change according with the various factors like smoking, age, diseases, etc. The process of keratinized it can be altered in cancerous and precancerous lesions like leukoplakia. There may be thinning of epithelium, changes in the salivary flow due to age, radiations, etc. The healing capacity of the oral mucosa it is greater than that of skin. So this was all about the oral mucosa.